हाँ बोलिए।
ओके सर, तो मैं स्टार्ट कर रहा हूँ। ओके,
ठीक है।
गुड आफ्टरनून डायरेक्टर सर, डॉक्टर सिद्धार्थ मैन एंड ऑल द faculty. Under the aegis of IQAC-ISFCP, today with us the expert in QSAR, Dr. Kabir Khan. He did his MS from NIPO Kolkata and PhD from Jadavpur University. Currently, he is working as assistant professor in the University of Gdansk, Poland. And over to you, Kabir. Please show your slides.
Is it visible?
No, it's not visible. It's visible.
Okay.
Yes, sir,
So,
visible.
okay. Thank you very much for this nice introduction. And talking about myself, I'm a postdoctoral researcher here in Gdansk. Okay, I have recently completed my PhD. So today I'm gonna talk about ecotoxicity modeling of contaminants of emerging concerns using QSAR approaches. Okay, I'll continue. So I'll be talking about CEC modeling using QSAR approaches. As some of you are already knowing what is contaminants of emerging concern. Basically, these are substances which has recently been discovered to possess environmental risk, and these substances were not actually taken very note of, like they were not being focused so far, and slowly they are being identified and found to be possess a lot of risk to the environment. So they are placed under one category called CECs. Okay. So since these substances were considered safe so far, so therefore no stringent policy was designed to estimate the toxicity of these contaminants in the environment. But in the last few years, the regulatory bodies, CD and ECA has devoted their attention towards these compounds, and we will see in the later like what are these compounds, which are those categories which actually poses like a CEC like compounds. Okay, so when you talk about which are the compounds which behaves as CECs, they include a array of substances. They can be from pharmaceuticals, household chemicals, personal care products such as cosmetics, pesticides, plasticizer, etc. So CECs basically constitute, they are present among all the chemical group constituents. Okay, so if you have to classify these CECs, they can be broadly classified into these different categories of substances. For example, among antibiotics, you have tetracycline. Among disinfectants, you have alcohols, etc. And similarly, the other categories you all can see on the screen. Okay, so now the question comes like if there is no test, we don't know the toxicity. So the researchers have now started to pay their attention towards these compounds. Okay, and they have tried to learn their degradation mechanism, how they have been accumulated in the environment, and which are the environmental bodies where they are affecting the most. Okay, so this is a rough estimate. It is assumed that there are approximately 100,000 or more chemicals are used, but they have very poor quality data. So we can actually claim something like with certain that these chemicals are actually responsible for something. Okay, so why they are important actually? Why we are talking about CECs? So first of all, we have to keep in mind that CECs, they are basically resistant to natural environmental degradation process. Why so? Because they mainly are composed of organic backbone, and as we all know, organic compounds are more persistent in environment compared to the inorganic one or the other compounds. Okay, so they can simultaneously accumulate and persist and cause a long-term or chronic response in the organism where they are exposed to. So the main target for these compound should be or is the aquatic bodies like river and their flora and fauna. Okay. So, as we are aware, like whatever contaminants are released into the environment, they are released into the water, and simultaneously they are being purified using wastewater treatment plants. But it is also a fact that these water treatment plants, they are not effectively purifying these compounds. Meaning they are ineffective totally of removing these compounds from the water bodies. Hence, they end up in rivers or oceans at the last age, and thus they pose a serious effect, serious effect, like they can go and persist in the environment and can have adverse effect on aquatic bodies, and indirectly to us also, because we are consuming those environmental animals actually. Okay, so this is one of the layout, like how these things are all connected. We utilize these compounds in the livelihood, in companies, in hospitals, and at the end they all go to waters, and they then are processed by sewage treatment plants and are being released into the oceans. Okay, but as I have already said, these methods have proven ineffective in actually removing or separating these compounds from the river bodies. Hence, the risk persists. Again, if you talk about the drug molecules, like they are what you can say, concentration, how much they are used across the world. The map is visible to all. These countries, they show some data, whereas the countries which do not have data, they are marked with light gray color. Okay, so as we can assume how much of the drug is released in these bodies annually, actually, and some of them are mostly in US, Canada, China, and India and Australia. Okay, so um, that's one good news though. We can actually determine acute concentrations, acute toxicity very easily in the labs. So because we can perform certain experiments in the lab in those targeted species. Okay, but as again I said, to determine chronic exposure, it's become a little bit difficult, because we do not know how much time it takes to accumulate in the body. So that problem still remains. Although the technology has developed now, so we have certain chronic endpoints calculation like NOAC and LOAC, but still they have not been proven to be very effective. So we need a better approaches to detect these contaminants into the environment. Okay, so I have listed some of the effects of these CECs. Most prominent and visible example is diclofenac. As we all are aware that diclofenac, apart from its properties, it has been responsible for killing several millions of vultures in Asia. Why so? Because the drug gets deposited into the bones and thus kills this species. So similarly, we have examples like oxazepam, which has a very what you can say, accumulating property in fish, etc. So as the moral of the story is, we cannot take these compounds lightly. Like we cannot simply release them into the water bodies. There has to be some way to remove them from reaching the environment. Okay, so this was already covered, like which are the sources for these pollutants. We all know that it's from garbage, waste, the plastic waste, etc., etc. It can be from industrial sources, it can be from hospital sources, and it can be from drugs which are being consumed regularly by the consumers, and also the agricultural source. Okay, this is another diagram depicting how much of the different type of drug molecules which are being administered in Germany only, not the other countries. Okay, so what measures we have come up with? Like scientists, what are the methods they have come up with? So basically, they perform these toxicity studies in environmental species, prominently in some group of species, not all, because it's next to impossible to carry experiments on all available species. So the scientists have identified certain group of species which can behave as a representative of that particular area or the surrounding. So most prominent among them is algae. Among algae, we have several number of species on which these acute and chronic tests are done. So the next category is bacterial species. Then crustaceans, among crustaceans, we have very common species called Daphnia magna, which is largely being used for finding the acute toxicity of these compounds. And then we have plants, fish, and mammalian cells also. We also find out the acute and chronic toxicity of these compounds in these cells also in vitro setting. Okay, and there's a protozoa also, and the list is very long actually. I have listed here some of those group of species and which are those species that are largely targeted. And tadpoles, of course, yes. Okay, so now uh, let's talk about persistence of CECs. Okay, what persistent is basically every organic molecule, since they are in organic in nature, they have the tendency to pass through the plasma membrane. So these can actually travel across the plasma membrane of the body of any species where they are encountered, and they can simply uh, cause a persistent effect. Like they can accumulate slowly over the years, and at the end, at the some particular time, they will have some adverse or hazardous effect on that particular species. And imagine if that particular fish or some marine environment is being eaten by the person. So these chemicals tend to transfer from that species to the mammals or to the humans. And similarly, this will start a cascade of transmission into the food chain. Okay, so we are not exceptional into this, so that persistence can happen to us also. Okay. Hmm. So another factor is biomagnification. Again, I've just explained what is biomagnification. They are increasing concentration of substances, like very high levels. They increase in the food chain. Okay, so it is very important to identify these substances, and not only identify, we have to prioritize them. How prioritize them? Like we can list them according to their threshold of causing toxicity or hazards to the environment. That is important. Okay, and these can be achieved in a very short span by using some modeling techniques. Okay, so now the major drawback is if you start doing experiments on one compound, it may take some time. If you start doing experiment on 5 compounds, it may take some more time. Similarly, this cannot go on for all the compounds because there are millions of chemicals and we literally cannot perform experiments for million of compounds. So what's the alternative? So we have these modeling techniques. So we try to develop models and try to predict the unknown compounds. So using these models, we can actually find out the fate of the substances and the exposure, like how much risk the particular compound or the CEC poses to the environment. Okay, so now this is the major drawback. Again, mainly in Europe and US, the regulatory bodies are now shifting towards non-experimental methods. Like these regulatory bodies are propelling scientists or toxicologists to avoid using animals for experiments unnecessarily. So why? Because there are several advantages of using computers, because it requires less money, less chemical waste, it is cost effective, and can be utilized very easily in the form of software or something. Plus, they are reliable and accepted. Okay. Okay, so now the first step of identifying CECs becomes substitution principle. So you may ask, what is a substitution principle? Simple. We try to substitute a more toxic compounds with a less one, which is having less prone toxicity towards the environment. This is the traditionally followed method. Like as the ages have developed in medicine, we know more and more safer products are being designed every day. One such example, there is a term coined recently called green pharmacy, which this deals with actually the modification of substitution of toxic compounds with the less toxic one. Okay, so one such example I have given here. This is one anti-cancer compound, ifosfamide, which is almost no biodegradable. It is highly not biodegradable at all, 0%. Whereas the second compound is near about 70% biodegradable. Okay, so such examples can be studies or can be studied for other compounds also, like other category of chemicals, in order to reduce the effect on environment. Okay, so talking about which are the models that are currently being used to study these environmental toxicity effect. So QSAR by far is the most popular technique across all the molecular modeling techniques, and this has been already implemented in several commercial softwares as a tool to identify these acute concentrations or chronic concentrations in the environment. And why they are accepted? Because they are being trained and tested on several species and proven to have effective to give beforehand the um, possible structural properties that are responsible for their toxicity. Okay, so how does the QSAR model looks like? Okay, it's basically a very simple description here. You can see BA is for biological activity, which is a function of chemical structure. So you may ask, what is a chemical structure or property? Chemical structure or a property is basically their attributes. Like, what are the attributes a chemical structure possesses? Name some. So I'll name some. For example, if you take one compound, say phenol, so it has a hydroxy group at one position. So that becomes it's one of the attribute. So it has a presence of hydroxy group. Similarly, there are certain theoretical descriptors which are being calculated using these structures using some softwares, which are in about 10,000 or so or more in numbers. One can find out if you want to see those. I have given here the link if you want to study more about the descriptors. This is in general the basic explanation of what a descriptor is. So QSAR equation basically tries to correlate the response, the activity variable with the independent variable, that is the descriptors, and check which are the descriptors responsible for this toxicity. Okay, so this is a overall flow of like how a QSAR model is developed. Basically, these flow is divided into 5 different OECD principles, and we need to follow this if we want to develop a model which is really helpful actually in this context. So the first principle for this QSAR model development is OECD principle one, which says there has to be a defined endpoint. When anyone is starting a modeling studies, he has to define his endpoint, like what he is working on, like what is the target of his motive, like what he wants to achieve. So he will be trying to model the lethal concentration of, say, pharmaceuticals in fish. So that becomes its defined endpoint. The second principle says there has to be some robust technique, like some techniques which are reproducible. It cannot be some random like which is non-reproducible, because if the technique is not reproducible, it's not science. There has to be a way to reproduce the results. Okay, so we have some training and test set division techniques available with us. Okay, and then there is a POICD principle number 4, which says there has to be some rigorous technique what we are using. It can be like there are many methods, we will be coming to that. Okay, and in this context we use the training set for model development and test set for validation. And OECD principle 3 says there has to be some applicability domain. What is an applicability domain? Applicability domain basically gives you the hypothetical space for the model, like if the model predicts something, whether it is reliable or not. We define that using AD. And finally, there has to be some mechanical interpretation, which actually necessary as part of OECD principles. Like we should be able to explain why these descriptors are responsible for increasing or decreasing the toxicity of that particular compound. So this is in brief, like how these QSAR methods or models are developed and what are the basic criteria to develop a QSAR model. And that has to be, if this has to be useful, these principles should be followed strictly. Okay, so what are the methods available in QSAR? Basically, this can be divided into 3 different types: regression types, classification type, and machine learning methods. The most popular among them for toxicity studies are regression-based method and classification-based method, whereas for other complicated equations or complicated problems, like where there is an involvement of huge data, machine learning methods are usually preferred. Okay, so among these three, the regression and the classification are termed as linear machine learning techniques, whereas the machine learning, the last part, is the non-linear. And first two are the classified, or you can say supervised techniques, And the last one is unsupervised technique. Okay. This I've already discussed, like what we need to have when we are developing any QSCR model. We need to have some standard concentrations available with us with uniform experimental conditions, and this has to be converted into some uniform units. And then there should be a span of at least 4 to 5 log units among the response data. These things are a little bit, what you can say, uh, more statistic. So you may have to read more about, to know about this. But these are the basic requirements what when somebody is taking up a molecular modeling task. Okay, this is how normal QSCR model looks like. Y on place of Y we have the response variable. A0 is the constant, A1 is the coefficient, X1 is the descriptor value. Okay. Okay. Then once the models has been developed, we need to validate those models. And these are some of the metrics I have reported here, which are used currently for validating this model. Like we have R square or the determinant coefficient, adjusted R square, variance ratio, etc. And similarly, for classification-based model, we have these classification metrics like sensitivity, specificity, accuracy, etc. This is called uh, confusion metrics. They are termed as confusion metrics. Okay. These already I have covered. They are 5 guidelines which we have to follow for developing QSAR models. Okay. You can see this book paper here, which describes in detail the process of QSAR model development by Dearden. Okay. And now let's talk about a little bit about read across. Now, since this QSR method was very popular, why there was a need to develop some more different new methods. So, read across, what is a read across? Read across basically is a terminology devised in the latest this decade actually. In many cases, QSR models basically fails to if there is a if your compound data set, like if your compounds are less in number, QSR model simply cannot read all the information from a molecule. So in those cases, the researchers have come up with a technique called read across. So read across have recently become very popular among those data sets wherein the data sets and their response are less. Like for example, if you are trying to model 20 compounds or 30 compounds or less than that, in that case, QSCR becomes almost ineffective. So we prefer the another technique called read across. These are of different types. One is analog approach, second is category approach. So you may ask, what are these approaches? So basically, they work on the grouping principle. We try to group similar compounds in a similar cluster, and we try to extract their meaningful descriptors, and we try to predict the unknown compounds. This is the simple explanation for a read across. And people have come up with several new methods employing read across, and I guess most popular among them is from my lab, Professor Kunal Roy, which is being widely used across the world. Okay, so far I have discussed about like what is CEC, how they are toxic and causing any effect to the environment, and what are the different modeling methods available to study these CECs. Now let's talk about a few case studies where these models have actually been used and demonstrated their application. Okay, so the foremost, we can see this paper. This was by me, and here we have tried to study the pharmaceutical compounds using QSCR and IQSTTR approaches. QSTR I've already discussed. In QSTTR approaches, basically we try to model response of one species against the response of another species. Thus becomes interspecies modeling. Another example, if I have to give, this is the another paper by jointly by me and my colleague Mohsin. Okay, here we have tried to study the biocides. Biocides are one of the group of compounds. They are mainly used in household for germ, in fact, in this microbes ke liye. Okay, and then this was another study by me. It was on endocrine disruptor chemicals, and we have tried here to identify several what you can say endocrine disruptor. Like if you all know what is endocrine disruptor chemicals, these chemicals basically when they are exposed to certain species, they cause disturbance in their endocrine system. So they may have effect on their reproduction, their gland, their hormone production, etc. Okay. And then there's another study which was conducted by my colleague Joyita Roy. She studied on earthworm using the same methodology of QSAR. Okay, and they have showed its effectiveness in identifying the structure properties which are responsible for targeting or causing acute toxicity in earthworm. And lastly, this is another study, okay, wherein they have shown the application of these QSTR or IQSTTR approaches in learning or identifying the structural properties of toxicity of these compounds against Dugacia japonica. This is another species, okay. So to conclude, we have so far we have seen the role of computational modeling of ecotoxicity endpoint along with the evolving areas of molecular designing approach for toxicity and risk management. The integration of in silico techniques with GC principles are needed not only for generating precise predictive models, but also for designing as well as synthesizing less hazardous and possibly non-toxic chemicals to reduce their environmental risk. So I would like to acknowledge my Horizon Europe and my university and National Science Center of Poland. Thank you all. And finally, these quotes for everyone. Essentially, all models are wrong because a model cannot be the right way, right thing, but some can be made useful. Your model has to be wrong, but that's okay if it is illuminating, if it is showing it, if it is imparting some knowledge to it. Thank you very much. I would be very happy to answer some questions.
Wonderful talk, Dr. Kabir. Now session is open for the discussion, and viewer can put their question in the comment section. I think there is no inquiry.
Mm-hmm.
Thank you very much, Dr. Kabir.
Thank you. Thank you very much. Thank you for having me.
Definitely, we will collaborate in the future.
Thank you, Dr. Kabir, for this wonderful talk. We highly appreciate your concern and your wonderful information and research work. Thanks a lot. Thank you so much.
You're welcome.